Welcome to the third lecture of week 5. Today I am going to talk about cell cycle division. So, as discussed in the last lecture, we saw that eukaryotic cells or our cells, they contain chromosomes. So, DNA is not a single molecule, rather it is divided into multiple molecules. So, these are chromosomes. Normally they are scattered like this. We normally arrange them according to their size. So, the longest one is chromosome 1 and the smallest will be 22, and then the sex chromosomes are named as X and Y. So, in case of cell cycle division or cell division cycle, we will see that segregating these different molecules, DNA molecules or chromosomes, into the two daughter cells very effectively is one of the most important challenges, and we are going to spend quite some time understanding how cells achieve this fantastic feat. So, let us look at these chromosomes once again. We have 46 chromosomes, right. So, we have 23 chromosomes, 1 to 22, and then the sex chromosome X or Y. Now, there are pairs of each. So, there are 2 copies of 1, 2 copies of 2, 2 copies of 22, and then X and Y. So, total is 46, right. So, this is called diploid. Now, it turns out that chromosome numbers have no apparent correlation to the organism size. So, this is us, this is a Reeves moonsack. So, it is basically a deer, has exactly same number of chromosome, but then this other deer, the Indian moonjack, it has only 6 chromosomes. Red ant, which is much smaller, has 48 chromosomes, more than us. On the other hand, blue whale, which is much bigger, has 44 chromosomes, so 2 less than us, right. Dogs have 78, so on and so forth, you can see. And this one, adder's tongue, so this is most probably the one organism which has the largest number of chromosomes. So, the number of chromosomes reported for adder's tongue is 1440, right. And you can imagine that when a cell has to divide, these chromosomes will be replicated. So, this 1440 will become 2880, right. And each of those chromosomes will have to be separated or segregated in the 2 cells, right. So, that is a fantastic task that is achieved by our cells every second. Okay. So, here is the structure of the chromosome. This is DNA. DNA is wrapped around the histone proteins to form the nucleosome, and then it is packed furthermore to form the chromosome, right. Now, we will see that normally this chromosome is something like this. In the interphase, when it goes into the M phase, it will become much more condensed. So, the pictures and diagrams that I showed you in the 2 slides back, that is normally in the M phase. So, this is again the histone protein, and the DNA is wrapped around this histone protein like this. Okay. So, what is cell division cycle, or in short, cell cycle? Cell division cycle is orderly sequence of molecular events in which a single cell duplicates its contents and divides into 2 cells. So, one cell will divide into 2 cells, and it has to distribute all its material correctly into the 2 daughter cells. This cell cycle of duplication and division is known as cell cycle or cell division cycle. So, what are the important features of eukaryotic cell division cycle? So, here I am going to discuss eukaryotic cell division cycle because eukaryotic cells are much complex and they undergo a lots of checks and balances, right. So, the first important feature is that there is a delicate balance between cell growth and division. So, if a cell continuously divides, then it will become smaller and smaller and smaller, right. So, if it becomes too small, then it will not be able to perform its task. So, after division, the 2 smaller cells will have to grow to a certain size before they can divide. So, there is this delicate balance between the cell growth and division. Cell division is required to form different tissues and organs. So, we are multicellular organisms, and if you typically think, there are all these different organs like your heart, lungs, liver, right. and each of these organs are made up of multiple cell types. So roughly there are 200 broadly categorized cell types, but there are many more. So these cells have to divide in order to maintain these tissues and organs. So one example that I can give you, and we will talk about this in more details in next week's lecture. So for example, if you think about your red blood cells, our red blood cells, they have a lifespan of around 120 days, which is around 4 months, right? So in 4 months, our red blood cell will die. So some will die earlier, some will die later, but that's the average time. So these red blood cells have to be replenished, and we have huge amount of red blood cells, right? So it turns out that our body makes 2 to 3 million red blood cells every second, right? So that's the huge amount of cell division that goes on every second that produces 2 to 3 million red blood cells, right? And that is needed to maintain a constant level of blood in our body, and that is true for most organs. So red blood cells is the most dramatic example. If you think, I will talk about our intestine in the next lecture, and you will see that when we eat food, our intestine is responsible for digesting that food and absorbing it. So it also undergoes mechanical wear and tear. So the lining of our intestine has to be replaced, and those cells divide every day, right? If you think about other cells, they will not divide that much, right? Now for cell division, there are two key components. The first one is the machinery which carries out the cell division cycle, and the second one is a control system. So this control system regulates whether it is a good time to divide, and then it goes through different phases. Whether the first phase is done, can it proceed to the second phase, so on and so forth. So there is this decision making process that is done by the control system, and the actual division is carried out by a set of machinery, and we will look at this in details. This machinery, we will see that it is microtubules, kinetochores and other things. These are rebuilt and disassembled every cell division cycle, right? So once the cell cycle is complete, they are disassembled, and when again a particular cell commits to go into a cell division, these will be rebuilt, right? And of course, cell division is tightly controlled by this control system, because if this control system is lost, then it can create diseases such as cancer. So the fundamental event in cell division cycle of living system is common. So whatever cell type we think, these are the common features. Duplication of the genetic material, because that is the most important part of the cell division cycle, and accurate distribution of the identical DNA into the two daughter cells. So first is duplication of the DNA. So all the DNA has to be duplicated. We have 46 chromosomes that will get duplicated to 92 chromosomes, and then each pair will have to be transported into the two daughter cells. So for example, if chromosome 1 has two parts, so one from the paternal side, one from the maternal side, and then there will be two copies of paternal chromosome 1. So one copy will go to one daughter cell, the other copy will go to the other daughter cell. Both cannot go to the same daughter cell, right? So this type of accurate distribution has to happen. The detailed molecular events of cell division cycle vary from organism to organism, right? Again, in a particular organism, depending on the cell type, it will also vary. Also, in this according to the stage of the organism. So embryonic cells will divide in a certain way versus the adult cells, right? So, but the overall features are common, and that is what we are going to discuss. So during early development, that is in the embryonic stage, cells divide once in 8 minutes. So they divide very fast, right? And in that case, actually what happens is we start with a large cell, and then it becomes smaller and smaller and smaller. And that is why you will see that these egg cells are so big, because they have to contain enough nutrient and enough material so that multiple rounds of cell division can happen. The epithelial cells in the lining of our intestine divide once in 1 day. Similarly, liver cells divide once in a year, and our brain cells, the neurons in our brain cells, do not divide. But this does not mean that the liver cells take 1 year to divide, right? So it means that their division is less frequent. These cells most likely take around 24 hours to divide. So, it will be very similar to these cells, but their frequency is much less. So, this is the overall schematic of a cell division cycle. So, there are two important phases in a cell division cycle. One is the S phase, here S stands for synthesis. In this phase DNA synthesis happens, and the other one is M phase. M stands for mitosis. This is where the segregation of the replicated DNA happens into the two daughter cells, right. So, these are the two important phases, and they are separated by two gap phases. The first one is called G1 and the second one is called G2. So, G1S, G2M, these are the four standard or important stages of a cell division. So, G1 is the first gap phase. It is a decision making phase where the cells decide whether to go through cell division or not. So, they will decide whether they have enough material to replicate DNA and so on and so forth, right. So, once that is done, it will enter the S phase, and in the S phase DNA synthesis happens. So, in our case 46 chromosomes will become 92 chromosomes. Then G2 phase is the second gap phase where various quality control measures are exercised. So, all the microtubules and all this machinery are assembled in this phase, and it ensures that the DNA synthesis has happened properly, all the chromosomes are present, there are no breakdown or damages in the DNA or the chromosomes, and once all those checks and balances are found to be okay, then the cell commits itself to the M phase. So, we will see, so I have already alluded to it, we will see that there are actually several checkpoints which ensure that everything is fine and the cell can proceed to the next stage. In many books, you will see that the cell cycle is shown like this, where the cell cycle is divided into two phases. One is the M phase and the other one is the interphase. So, G1, S phase and G2, these three are combined as the interphase, because in this the most important event that happens is the DNA replication, and the other phase where a lot of action happens is the M phase. So, we are going to go through this M phase in more details now, because we have already covered DNA replication in earlier lecture. The key components of the machinery of cell division cycle are as follows. The first one is microtubule. We have already seen microtubule, which is a polymer of proteins, right. So, we have seen that tubulins are the monomers of microtubules. A tubulin dimer forms this hollow tube of microtubules, which is a part of a cytoskeleton. The next one is kinetochores. These are complex structures formed from proteins on a mitotic chromosome, to which microtubules are attached. So, these microtubules get attached to these kinetochores, and they help in the movement of the chromosome to the two poles, like so. You can see that chromosome movement is shown here, right. Then there are centrioles. These are short cylindrical array of microtubules. These centrioles are at the two end of the cell. So, they form the two poles, and then there is contractile ring. So, this ring contains actin and myosin, and they contract to pinch the two daughter cells apart. So, this pinching that happens, this is because of this actin and myosin cytoskeleton proteins. So, let us look at the different phases of the M phase or mitosis. The first one is prophase. So, in this case the chromosome condenses to form sister chromatids. So, you can see that there are two chromatids, right, they are connected together. Now, remember that DNA replication has already happened. So, these two are the exactly same chromosome, there are two copies of that. So, this is inside the nucleus. Outside the nucleus, centromeres have replicated and moved apart. So, initially there was one centromere, they have replicated and now they are moving apart. So, they are moving apart, and these green lines that you see, these are microtubules. So, they push each other, and that results in these two centromeres or centriosomes moving apart. This is an actual fluorescence image. In this case, the DNA is colored red. So, that is a red dye, and the microtubules are colored green because they have bound to green dye. So, we will see these two images for all the different stages, right. Now, mitotic spindles assemble between the two centromeres. So, these are the mitotic spindles. In the next phase, which is the pro-meta phase, the nuclear envelope disintegrates. The envelope is gone. Now the chromosomes are exposed and they can get attached to these mitotic spindles. So they get attached to the mitotic spindles through their kinetochores. And now the chromosomes undergo active movement. So they will get pulled and pushed around this inside the cell. In metaphase, the chromosomes are aligned equidistant from the spindle pole. So this is something that is very important because the cell has to ensure that these two sister chromatids, one goes to this side and the other one goes to this side. So to ensure that all the sister chromatids are arranged at this plate at the center of the cell. Kinetochore microtubules attach to these sister chromatids to the opposite poles of the spindle. So this sister chromatid is attached to this and this sister chromatid is attached to this, right. So they will grow and shrink. So this push and pull will happen and that will ensure that all the sister chromatids are at the center of the cell. The fourth stage, which is the anaphase, triggers suddenly, and that happens when these two sister chromatids are separated. So how this happens? This happens because the sister chromatids are connected together by certain proteins called cohesins. So you can think of them as glues, right. So these cohesin proteins, they form a complex which attaches these two sister chromatids together, right. The change or the beginning of the anaphase is triggered by the degradation of these cohesin molecules or cohesin proteins. So there are proteases which will get activated at this cell, at this phase of the cell cycle, and they will degrade the cohesin, and once that happens, this glue is gone and the two sister chromatids are separated. Now these microtubules, they start shortening. So shortening of these microtubules result in moving of the sister chromatids towards the two poles. So 46 chromosomes will move in this direction and the remaining 46 chromosomes will move in this direction, and this ensures that exact distribution happens. So the two paternal chromosome one will go here, the other one will go here. The two paternal the two maternal chromosome one will go here, the other one will go here, right. So like that. So in the last phase, the daughter chromosomes arrive at the poles. So they are here. Now they will start decondensing. New nuclear envelope starts building. You can see that the nuclear envelope is building. So there will be two nucleus, right, and that marks the end of mitosis. Now what remains is the division of the cell. So that is cytokinesis. So the cell membrane will start invagination like this, and that is triggered by the contractile ring that is formed by actin and myosin filaments. So we already saw microtubule filaments. Here actin and myosin filaments become active. So they start forming a ring here and they will start contracting. So that will pinch the cell membrane and finally result in the formation of two daughter cells. And you can see there are microtubules and each will have one centriole each. So that is the cell division. Now remember that what we have done here so far is we have looked at the division of the genetic material or segregation of the genetic material. But there are other cellular organelles which are also present and they also have to be efficiently divided into the two daughter cells. So that is done by these three different mechanisms. The first one is for abundant organelles. Organelles like ribosomes, peroxisomes, which are present in large numbers. So ribosomes will be scattered throughout the cell, right? Now if the cell divides like this, you will have enough ribosomes in the two daughter cells. So it's just by sheer number they are distributed into the two daughter cells. Chloroplast and mitochondria, these are very important organelles. So to ensure that both cells get enough of these organelles, they divide by fission, just like bacterial cell division, okay, and they are transported by motor proteins along the microtubules to the two daughter cells. So they sort of hitchhike on these microtubules along the two poles. That ensures that enough chloroplast and microtubules will be present in the two daughter cells if it divides like this. Golgi and endoplasmic reticulums, they divide into smaller vesicles, and then during cell division, they are segregated into the two daughter cells. After the division is complete, they fuse together to reconstitute the intact Golgi and endoplasmic reticulum. So, this is a recapitulation of what we have done so far, that we have 46 sets of chromosomes, it becomes 92, and then it gets divided into two cells, which is 46 and 46, right. So, this type of division happens in cells which are called somatic cells, which are diploid, because they have two copies of each chromosome, one from each parent. There are other types of cells that we have, which are the sex cells or the gametes. So, sperm and ovum, these are haploid, right. So, they have only one copy of the genome. So, the sperm and the ovum, they will have only 23 chromosomes, right. So, how does this happen? So, how do we get these cells? What division is responsible for that? And we will see that it is done by a different type of division, which is called meiosis, right. So, this is just for your information that if there is one set of chromosome, it is called haploid. If there are two sets of chromosomes, it is a diploid. We can have 3, 4, 6 and 8 cells, right. So, strawberries are an example of octaploid, where they have 8 sets of each chromosome. So, strawberry they have 77 chromosomes, and there will be 8 set for each of those chromosomes. So, 7 times 8, 56 chromosomes are present in a strawberry. Okay, I will come to this, how this haploid cells are generated, but the fertilization of two haploid cells, so sperm and ovum, they will have, so in our case this will have 23 chromosomes, this will have 23 chromosomes. So, when they come together, fertilization happens, and then embryo will have 2n sets, because 23 comes from here, 23 comes from here. So, that is total 46, right. So, how do we get these haploid gametes in a diploid organism? So, before I go into that, this is just an overview of a life cycle of an organism like us. So, life starts as a single cell. So, it is a zygote, it is a diploid. So, there are two sets of chromosomes. Now, mitosis will happen, right. This is the normal cell division that we have seen, and all these different cells will be generated. Each of them will have 2n copies of DNA. And then something interesting happens, which we will discuss in the next lectures, that differentiation. So, the cells, these cells are most probably all very similar to this, but then the cells will differentiate. So, there will be skin cell, there will be different organs that will develop, there will be bones, right. So, all these different cells are generated, and that is also called morphogenesis. So, the body plan happens. So, the body is developing, right. So, we will have almost 200 different types of cells. Now, all of these are mitosis. So, mitosis will actually generate different types of cells, right. And then we also have to generate the sperm and ovum, which are generated by meiosis. So, in this case 2n is maintained during mitosis, and meiosis will result in the formation of n. So, 23 chromosomes here, 23 chromosomes here. Now, these two combine together to form again a new life, which is 2n cell. So, meiosis undergoes two divisions. So, this is a 2n cell, it DNA replication happens. So, it will be 4n, and then it undergoes first division. So, this is 2n and 2n, and then it will undergo another division. So, each of these will be n. So, from one cell we get 4 gamete cells, right. So, it can be sperm or ovum, and we end up with cells which are n or haploid. So, the same thing is shown here, right. So, mitosis is equal division, 2n goes to 4n, and then from 4n we get two daughter cells, 2n and 2n, right. So, this is for diploid. If an organism is not diploid, but again mitosis happens, then it will be n goes to 2n, and then it becomes n and n. Meiosis is reduction division. So, in this case the number of chromosomes will decrease. So, 2n becomes 4n. The first division will result in two cells, each will be 2n and 2n, and then the second division will result in 4 cells, each of them will be n, n, n and n. So, here is a comparison. This is mitosis, we have already seen this. So, let us focus on meiosis, right. So, in this case we have taken a very simple cell which has only one chromosome, right? So, one chromosome, now there will be two copies, one from the father, one from the mother. So, paternal and maternal, right? So, first step is DNA replication. So, now there are two paternal chromosomes, you can see two red chromosomes here and two maternal chromosomes, two grey chromosomes here. So, these are the sister chromatids and they are connected together by the cohesin molecules, right? Now, something interesting happens. So, in case of mitosis, these chromosomes are arranged like this, okay, and each of these will be connected to the two poles on the different side so that they can be distributed like this. Here they are arranged side by side and there is something that is called crossing over. So, you see that this is chromosome 1. So, red one is from the parent father, grey is from the mother. Now, there is connection between them. We will see this picture in more details in the next slide. In the first division, what happens is this is 2n, but both sister chromatids have gone in this and both sister chromatids have gone in this cell, right. So, these two cells are not similar to these two cells, okay. Now, another division happens which is similar to this and the two sister chromatids go into two different cells. So, we end up with 4 cells, right, and each of these will have only one chromosome 1 and that can be from the paternal side or that can be from the maternal side. However, there is some mixing that has also happened which is called crossing over, because you will see that a small bit of red has come here and small bit of grey has come here, right. So, this is something that is called crossing over and that results in genetic diversity. So, in case of mitosis, the two sister chromatids are attached like this by chromosome by the cohesin molecules or cohesin proteins. Kinetochores are here and they are connected to the two poles in two directions. So, they will get separated here. So, one will go here, the other one will go in this direction. In case of meiosis, the two sister chromatids are present side by side. So, this is from the paternal, this is from the maternal, and both are connected on one side and here both are connected on the other side, and here crossing over happens. So, some part of this chromosome will go here and some part of this chromosome will go here. So, the same thing is shown here in more details. So, you can pause the video and just go through this yourself. So, in this case what you will see is the first division results in these two, right. So, both maternal chromosomes are on one cell, both paternal chromosomes are on the other cell, and then the second division will result in 4 daughter cells, each are haploid, and you will see that some bits and pieces of the maternal chromosome is with the paternal chromosome and here the paternal chromosome is with the maternal chromosome, right. So, this DNA is not exactly the same as this DNA, right. So, this is genetic mixing, okay. But nothing like that happens in meiosis. So, whatever is here, that is exactly what you get here, right. So, no mixing of DNA happens. Mitosis, it ensures that every cell in an individual carries the same chromosome number, same genomic content and same biological information. So, mitosis is genetically conservative, right. On the other hand, meiosis is reduction division. So, it ensures that species specific chromosome number remains constant over generations, right, because reduction division happens and then the sperm and ovum will combine. So, it will again give rise to 2n, right. It also contributes to genetic diversity that stimulates evolution. So, this is a very important part that it plays, right, because random segregation of chromosome in the gametes and crossing over results in exchange of material between the paternal and the maternal chromosomes. So, a child will always have some genetic material that comes from the father and some genetic material that comes from the mother. Okay. So, we have seen how cell division happens and how the machinery plays an important role. Now, we are going to focus on the control of cell cycle, cell division cycle. So, this is just a recapitulation of what we have seen: G1, S, G2 and M, right. So, these are the 4 phases. So, a very elegant experiment was done almost 50 years back by Rao and Johnson, where they figured out that this cell goes through these different phases and there are checkpoints, right. So, what they did, they took two eukaryotic cells and they fused them, right. So, I am not going to go through the technology, but let us see what is the outcome. So, one cell was in S phase, right, means it is replicating DNA. The other one is in the G2 phase, which is after S phase, right. So, when they fuse them together, so this one cell will have two nucleus, right. Now, the question was, can they exchange material between each other and can they influence each other? So, you would expect that, okay, this is in S phase, this is already done, so it can proceed to the M phase, but that is not what happened. This somehow retarded the progression of G2 into the M phase. Once this is done, DNA replication is completed and it goes into the G2, they both together go to the M phase. So, it means that whatever factors here can diffuse into this nucleus and it can inhibit it from going into the M phase, right. So, there is some sort of check that once both are into the G2 phase, all the DNA replication has happened, then only the cell went into the M phase. They did other sets of experiment. So, here they fused S with G1. So, in this case S with G1, right. So, what happened is that G1 did not prevent the S phase, but the G1 went into the S phase very quickly, right. It means that there are factors in this which induced this nuclei to go into the S phase quickly. They also fused G1 with G2. So, one cell, one nuclei in this and one nuclei in this. So, in this case G2 nucleus had no effect on G1. So, G2 did not have any effect on G1, but G1 delayed the entry of G2 into the M phase, right. So, only when G1 got into G2, then only they together progressed into the M phase, right. So, this means that there are checks and balances at every phase of the cell cycle which retards the next step until the current step is completed. So, that is the check phase, checkpoint of a cell cycle. So, it turns out that there are three major checkpoints. The first one is the start or restriction point, right. So, start or restriction point is here. So, after the G1, the cell decides, is the environment favorable, do I have enough material to produce replicate DNA, because the DNA number will double in the S phase. Then there is this G2M checkpoint. So, it is between the G2 phase and the M phase, right. So, the cell has to decide if all the DNA is replicated, right, is the environment favorable, all the machinery is there, now it can enter the M phase. And once it is in the M phase, there is a checkpoint between the beta phase and the anaphase. So, at the meta phase, remember that all the sister chromatids are arranged at the center plate, right. So, they have to be properly arranged at the center plate. Once that is a center plate, once that is done, then only the anaphase will be triggered, because the cohesin molecules will be degraded and the two sister chromatids will be pulled apart along the two poles. So, what is the mechanism of these checkpoints? How does this work, right? There are proteins which are called cyclins and another set of proteins which are called cyclin dependent kinases, which are the central component of this cell cycle control system. So, you have already seen kinases. So, kinases are enzymes which phosphorylate certain amino acids. It can be a tyrosine residue, it can be a serine or a threonine residue, right. So, these kinases will not do that unless the cyclin is present. So, for example, this is a kinase, it will be inactive. Once this cyclin comes in, it will bind this cyclin dependent kinase and it will get activated. So, it will phosphorylate certain proteins that will trigger this set of events. Once its job is done, the cyclin is degraded, which means that the kinase is again inactivated, right. So, different phase of the cell cycle is driven by different cyclins. So, we will see that different cyclins are produced at different phase of the cell cycle. So, it is shown here. There are three classes of cyclins that are required in eukaryotic cells. The first one is the G1 to S cyclin, which is shown here in blue, right. So it binds to the CDKs or the cyclin dependent kinases at the end of the G1 phase and commit the cell to DNA replication. So this cyclin is produced, it will trigger this G1 to S phase, it will trigger the cell to go from G1 to the S phase, and then it gets degraded, right. Now the second cyclin comes in, which is the S cyclin. S cyclin binds to CDKs during the S phase and is required for the initiation of the DNA replication, right. So it will be present throughout this, and then it will get degraded. And then the third one comes in, which is the M cyclin, which promotes the events of the mitosis, right. So it is produced here, and then again it is degraded, right. So these cyclins, their production and degradation will drive the cell in a particular direction. That is why cell cycle happens in this unidirection, right. Okay. So again, going back to this, we know how cyclins are produced, right. So we have already gone through this. Gene expression happens, mRNA will be produced, from that protein will be produced. So we can control the production of protein by controlling the production of the messenger RNA, which is transcription, right. But how do we ensure the protein is degraded once its job is done? Because proteins are quite stable molecules, right. So there are active components inside the cell which degrade proteins, right. So take for example this cyclin, M cyclin. Once its job is done, it will get tagged by certain molecules or certain proteins by a certain molecule called ubiquitin. So ubiquitin gets tagged onto this, and once that happens, then protein degradation machinery, which is called a proteasome, it will know that okay, this protein is marked for degradation, so it will catch that protein and it will degrade it. So that is shown here. So this is a protein which is tagged by ubiquitin. So ubiquitin is a very conserved molecule. It consists of 76 amino acids, and it binds to the lysine of the protein. So the lysine side chain of a protein which gets to be degraded, right. So on that ubiquitin is marked. Now the proteasome will know that ubiquitin, this protein has to be degraded. So it will bind that, it will denature it. So using mechanical force, it will denature the protein. It will strip off the ubiquitin because you don't want to degrade that ubiquitin, you want to recycle it, and then it will chop up this protein into smaller fragments, smaller peptides, right. And then those peptides will get degraded by other proteases into amino acids, and those amino acids will be recycled to form new proteins. So here are some of the features of cell cycle control, right. So it's an interesting theme in which molecular events of cell cycle control that we have seen. So here each phase activate the next step, right, and in that particular phase, and also prepare the cell for the next phase of the cell cycle. So a current phase will prepare the cell for the next step, and once everything is favorable, it will move the cell into the next step. So the sequential or properly ordered events are maintained in cell cycle. Partial or complete loss of this cell cycle can result in diseased condition such as cancer. In normal cells, minor problems such as DNA damages are repaired. So there are proteins which continuously ensure that there is no DNA damage that is present. If there is present, then they will repair it, and it will halt the cell in that particular phase. Once all those damages are repaired, then the cell will move on into the next step. If the DNA damage cannot be repaired, then the cell will trigger a particular event which is called apoptosis. So it is programmed cell death, right. So you don't want that damaged cell to be present in the organ, you want to eliminate it from the organ, right. Now several defects in the cell cycle checkpoints may lead to abnormal or faulty molecular events. So accumulation of multiple mutations and DNA rearrangements in the genome can result in disease phenotypes such as cancer. So understanding the detailed mechanism of this cell cycle control can help us to develop treatments for these different diseases. So these are the books that you can refer to. So the first two are very important. You can go through these books for today's lecture. Thank you.